everyone and welcome to Instant Biology by Dr. Neelav. The topic that we would be dealing today is RT-PCR or Real Time PCR. Let us be sure about this. RT-PCR is sometimes also said that it is reverse transcriptase PCR, but we here are dealing with Real Time PCR. Okay. So for those who have been following me on my YouTube channel, they would have seen a lecture on PCR already. That lecture actually was meant to discuss about the basics of polymerase chain reaction. Now in this series we are aiming to address different types of PCR such as hot start PCR, such as nested PCR and real-time PCR, reverse transcriptase PCR. We would be dealing with each of them in due course of time. I have started with this real-time PCR. Okay, so as always I have divided my lecture into the following points, and we would deal with each point in detail so that you can understand this in a better way. So let us start with the first point. It says that this real-time PCR is also known as kinetic PCR, qPCR, qRT-PCR, RT-qPCR. These are the names of real-time PCR. Now why is this called kinetic PCR? Because you might have heard about kinetic energy, about the movement it involves in. Okay, so here what do? We are analyzing things in real time. We are doing it currently. What is happening in the current scenario, we are interested in that. That is why here kinetic word is used, and in most of the words you can see that Q has been utilized, Q has been used. Now what is basically Q? Q is quantitative PCR. Here we try to determine that how much quantity of a product is being made or how much quantity of a gene is being expressed, right? So that is the meaning of Q. Okay, now let us go on to discuss about copy number. That here we have written that it is utilized. Basically, what is the need of real-time PCR? So the need of real-time PCR is to determine the copy number or to determine the gene expression in real time. What does this mean? How much of a gene is being expressed? What is the copy number of a gene, that how much time it is being expressed? We need to know this, or we need to know this, that how much gene is being expressed. You would be knowing that the DNA constituent in a cell is same across the whole body, but some genes are expressed in particular organ and some genes are present and some genes are expressed in another organ. Suppose you are aiming at the expression of a particular protein that is involved in visualization, so it would be expressed in your eyes and it won't be expressed in your liver. Okay, so we want to know that how much a gene is being expressed, and we do that with the help of real-time PCR. So this would be clear to you now that we intend or we aim at determining the copy number or gene expression in real time. You need to understand this. RT is written here, which means real time. Real time is just now, currently, right? So in most of what does real time mean? In most of the PCR reactions, in most of the PCRs that you would have seen, what happens is you just get to know about the result of the PCR reaction after the completion of the PCR cycles. Once you put in your tubes inside the PCR machine, after a due course of time, after 2 hours or 3 hours or whatever program that you have fed inside the PCR machine, after that only you come to know that what was the result of your PCR. You then run your PCR products onto an agarose gel, and then you come to realize that was your product amplified or not. You come to know about that. Before that, if you want to know, you cannot know. In no case you can know that your product has been amplified or not in the middle of the PCR reaction. But this is where real-time PCR is different. You can monitor the reaction in as it goes along. You can monitor it as it goes along. So this is the very uniqueness of real-time PCR, right? So in the next point, what do we say here is initial amount of template. Now, what is our aim? I already said to you that we want to know that how much of a gene is expressed in the sample. That is what we want to know. So in real-time PCR, what does real-time PCR aim at? It aims at judging or knowing the initial amount of template, right? So you would be having a template, you would be having a particular amount of gene or product that was being expressed. You need to know that how much gene was expressed. You need to know that. So if you consider that when the real-time PCR is run, the products would be obtained in proportion to the initial amount of the gene expressed, initial amount of the gene that was expressed. If the initial amount of the gene that was expressed was high, the resultant product or amplification would be more. If the gene would be less, if the gene expression would be less, then of course the product amplification would be less. So this is the concept that you need to understand before knowing about the real-time PCR. So what do we aim at? We aim at knowing the initial amount of template. How do we judge that? We determine the end amount of the product that is being formed, and with the help of a formula we come to know that what was the initial amount of the gene that was expressed. Okay, so we would deal with the equation in some time, but I guess this initial introduction of the real-time PCR would be clear to you by now, right? So this is what I wanted to say about the introduction of real-time PCR. Now we move on to the instrumentation. What all instruments or what all components are involved in determining or in the running of real-time PCR. The first one is thermal cycler. It is again, you would be knowing that a thermal cycler is a PCR machine. Next is computer. The thermal cycler is attached to the computer for the purpose of computing, analysis and everything. Next, optics, fluorescence emission and absorption. Now you need to understand here that a dye is involved over here. What is a dye? It is a it is a substance that gives emission, or that is absorbed at particular wavelength. So that particular dye is mixed in this reaction. Okay, so as the reaction continues, as the PCR is running, we analyze or we determine that how much of that particular dye is emitting the fluorescence, and we come to know, we calculate that, and with the help of the emission that we have obtained, we determine that how much of the product is being made. We will understand this in some time when we discuss these steps, but for now you need to understand that an optics instrument is also involved in this, which has fluorescence emission and absorption part, right? So now last is data acquisition and analysis. So this is the main part, actually the main part of the instrument which handles the data acquisition. What does data acquisition means? To intake the data, to take the data from the thermal cycler, and analysis of the data that has been obtained. So this was the last component of the instrumentation. Now coming to the principle, on what principle does the real-time PCR work? Okay, so the first point is detection and quantification of a fluorescent reporter. So as I've already told you that a fluorescence reporter is being used, that the one which I called as a dye. It can be called as a reporter. Why? Because it reports to us that how much gene is being expressed. That is why it is called as a reporter. So what do? We determine, we detect and we quantify that how much fluorescence reporter is present. How do we do that? Because the reporter has the property of emitting fluorescence. So we have this optics instrumentation which would of course tell us that this much fluorescence is being generated. Now with the help of that, we would come to know that this much of reporter is present. Now when this much of reporter is present, definitely the reporter would have bound to the product, the PCR product. So we can guess that this much of the product was formed, and if we know that this much of the product is formed, then we can assume, we can come to know that how much gene was expressed, or how much of the product was initially present. So this is the basic idea of this real-time PCR. You would understand it more as the lecture progresses. Next is first significant the first significant product in the amount of PCR, the first significant increase. It determines that how much increase was there in the first cycle, and which leads to determining the initial amount of the product that was present. It would lead to the determination of the initial amount of that particular gene that was expressed. So that this is what the principle actually is. Now let us come to the reaction profile. Okay, so let us divide the reaction profile into 3 steps. Okay, so the first step is early background phase. Okay, let us understand this by drawing a few diagrams over here. Okay, so let us understand this early background phase. Okay, suppose we have a dye. Let us say that we have SYBR Green as a dye. What is the property of SYBR Green? SYBR Green has a property that it would bind only to the double-stranded DNA and no other form of a genetic material. It won't bind to any other form, but it would only bind to double-stranded DNA, right? So what will happen is, suppose that you have a PCR machine, you have a tube, say you have a tube. In this tube you have put your DNA template. Next you have put your DNTPs. Next you have put your Taq polymerase. Next you have also put your this SYBR Green. We would deal with the method of working of SYBR Green in more detail as the lecture progresses. You have your SYBR Green. You have let us consider that we have all the material that is present inside this tube. Okay, so once the PCR starts, what will happen is, as the reaction proceeds, you reach to around 92 degrees centigrade. What will happen is the DNA would denature. The DNA would denature, and I have already said what does denaturation mean. The hydrogen bonding between the 2 strands would break. Okay, when the hydrogen bonding will break, what will happen? The double-stranded DNA would convert into single-stranded DNA. Right? The double-stranded DNA would convert into single-stranded DNA. Now I have already told you that this SYBR Green won't bind to single-stranded DNA. This would only bind to double-stranded DNA. Okay, so in the first case, what will happen? In the first case, because the DNA is in single-stranded form, would this SYBR Green bind to this single-stranded form? No, it won't bind. Therefore, the SYBR Green will give fluorescence only and only when it is bound to this double-stranded form. When it is in the form of unbound form, when it is not bound to the double-stranded DNA, let us consider this. When it is in the bound form, then only it gives fluorescence. When it is in the unbound form, it won't give fluorescence. Okay, and where does this SYBR Green bind? It will bind to the double-stranded DNA. So in the initial phase, because the denaturation has occurred, it will have only single-stranded DNA. So most of the SYBR Green would not bind to this. Therefore, the fluorescence would be very, very less. The fluorescence would not be more. The fluorescence would be very, very less. So this is what we have written, that In the early reaction, it would be in the background phase. Background phase means it would be the fluorescence would be just like background noise and no detectable fluorescence would be present, right? Understood? I guess this would be very clear to you. In the first phase, no detectable fluorescence would be present, and this is called as early background phase. Now coming to the next step, what happens next? Because the DNA has been broken down into single stranded form, what will happen? The primer would bind here, the primer would bind here, because we are starting with annealing, right? I request to the viewers that those of you who have not seen my PCR basics lecture, I would paste the link in this video. Kindly go and visit, kindly go and see that PCR video, then this would be very clear to you. So the next step would be annealing. What does annealing mean? The primer would bind to the template because we need to amplify. The primer has bound to the template. Now this strand is in the double stranded form, right? What will happen is and the elongation has started. What will happen is the primer has bound and the elongation has started. Annealing, and we are talking about the annealing and elongation phases. What will happen? The primer has bound and started to elongate. Now what will happen? This strand is now double stranded. I've already told you that SYBR Green would bind to only the double stranded DNA. Now when the double stranded DNA has started to form, what will happen? SYBR Green will bind to it and start to give the fluorescence. That is why we have written here is fluorescence increases rapidly. Why? Because the cycles are continuing. Why? Because the cycles are continuing. The DNA, the Taq polymerase is working. It is forming the other double stranded. The products are being formed. That is why the fluorescence increases. Okay, this would have been clear now. Now there is one more concept about CT or threshold cycle. Now what is threshold cycle? I have written it. You can write down the definition also. Threshold cycle are the fractional cycle number at which the fluorescence passes the fixed threshold. Now suppose we have a threshold below which the fluorescence is not detectable. In this case, the fluorescence was very low. It was below the detection levels. So it is that number of cycles, that CT or threshold cycle is that number of cycle which is required to pass this detection limit. This detection limit is crossed at that particular. Suppose after 5 cycles or 10 cycles, so much product is accumulated that SYBR Green starts to fluorescence in a high amount, and this detection limit is crossed, which is that particular cycle is called as threshold cycle or CT. Okay, so this was CT. Now there is a formula with which you can determine that what was the initial amount of template. With this you can come to know that what was the initial amount of template. Suppose in this formula Tn is equals to To e to the power n, right? Now let us understand what are the parameters here. Tn, with Tn you can say that Tn is the amount of target sequence at cycle number n. After n cycles, after n number of cycles, what is the amount of the target sequence? Suppose you were amplifying this particular sequence, okay, this is To, the amount that is present initially. Then after n number of cycles, what was the amount that was present is Tn, and E is the efficiency of amplification. So each PCR has its amplification efficiency. So E is the efficiency of amplification, N is the number of cycles. Okay, after n cycles we are only coming to this particular equation. Okay, after this, the last we had said that the reaction profile was divided into 3 steps. The first step is early background phase. The second is fluorescence was increased to rapidly in the elongation or the annealing phase, and next finally what happens is the fluorescence starts to decrease, and because after some time the PCR cycle would cease. Why would it cease? Because the efficiency of Taq polymerase slows down as the reaction progresses forward. After a particular, suppose say 40, after 40 cycles, the efficiency of Taq polymerase goes down and the reaction slows down or stops. So what will happen is the decrease is seen, decrease until no product accumulates, decrease up till decrease until the no product accumulates. After the product stop to accumulate, what will happen? The fluorescence will of course decrease. So this was the reaction profile. Now coming to the SYBR Green, let us understand that how SYBR Green chemical tells us that a particular gene is expressed or not. First of all, what happens, I've already told you that this SYBR Green has the ability to bind only double-stranded DNA. It won't bind to any other form of DNA. It will just bind to double-stranded DNA. Fine. Now, denatured. Now, in the first case, what will happen? Because the DNA has been denatured, I've already told you in this portion that DNA was denatured, as we all know. Denatured DNA plus primer and plus the dye, right, is present. Because the DNA here is denatured, therefore what will happen is the SYBR Green won't be able to bind here, the fluorescence would be very less. Next, annealing phase, the fluorescence starts. Next, elongation phase, the fluorescence increases rapidly in real time. You can just observe this as it happens and as it progresses in the reaction. Next is denaturation. After denaturation, what will happen in when the cycle is completed? Denaturation occurs because it again the cycle again continues from 92 degrees centigrade. The two strands separate and then signal again falls down. Let us understand one more thing, that the DNA that we are talking about, that we have talked about in this entire up till this point, is cDNA. You need to understand this, that we were talking about cDNA, right? cDNA is obtained from the mRNA. Okay, you should be clear about this, that we are not talking about the DNA, the normal DNA strand. We are talking about the cDNA. cDNA is obtained from mRNA. mRNA is the RNA that has been produced in a particular tissue because it needed that particular genes to express. Now that mRNA has been changed into cDNA. So now we have the complete information of that genes, of the genes that were about to express in a particular organ. We have the information in the form of a cDNA. So we are working the entire things that I have told you, we are working on cDNA. Please do not confuse with the normal DNA. Okay. In the last portion of this real-time lecture, I would like to introduce you to a graph. In this graph, as I've already told you, that a baseline is a threshold, or a baseline is this threshold is described, right? This is the threshold. Okay. So below the threshold you cannot detect, actually below the threshold you cannot detect the fluorescence. This is how the PCR proceeds. This is the how the PCR reaction progresses. These are the values, that is 20000 DRN. These are now let us these are basically the increment of the fluorescence signal. After each cycle, the fluorescence signal increases. Why? Because each time more products are formed, more double-stranded DNAs are formed, so more SYBR Green would bind to them. Therefore, DRN means increment of the fluorescence signal. This would be clear to you. And baseline is PCR cycles. After a number of suppose after 5 cycles, the fluorescence has not reached up to a level that it can be taken, or it can be it can be acquired, or it can be assessed, or it can be determined. Okay, so those number of cycles would come under the baseline only. Okay, suppose 5 cycles, 6 cycles. After suppose 10th cycle is that which causes the signal to be higher than this threshold, this particular point at which this cycle, suppose 10th cycle, after 10th cycle the signal becomes high enough to cross this threshold. That particular point is called, or that particular cycle is called as threshold cycle. We have already talked about this here, so we are just discussing it in the form of a graph over here. This is the exponential phase. Of course, the elongation phase, the annealing phase and the elongation phase, this is in that portion, exponential phase. I've already told you about threshold, I've already told you about baseline. So these are here in the x-axis. We are taking the number of PCR cycles. Suppose after 20 PCR, here after 20 PCR cycles, what is happening after 20 PCR cycle? The threshold is being crossed. So here 20 is the threshold cycle. Okay, I guess you would have understood the concept. Here after this part there is no amplification. Why? Because the effect or the affinity, basically the, what do you say, the effect of the Taq polymerase goes down significantly. Why does this happen? Because the tube becomes so much crowded, there is so much of product, there is so much the turbidity in the tube that the function of the Taq polymerase becomes hampered. So this was basically the graph related to the real-time PCR. I hope this lecture would have been beneficial for you and you would have understood the working of real-time PCR. I would insist that you please go and watch the normal PCR lecture, and I have also formed a Telegram group, and I would definitely post the link to my Telegram group in the description box. If you have any issues with this lecture or any other lecture, you can post it in the comment box and I would love to solve it in the upcoming lectures. If you like my videos, please hit the like button and subscribe to my channel. Thank you so much.